Hello, listeners. Yamina here. I'm very excited to have you join us for another Dr. GPCR podcast episode. If you haven't heard, we are officially a nonprofit 501c3 organization. Becoming a nonprofit only solidifies our mission to serve the GPCR community. Therefore, we're taking a short podcast break starting in July to come back with new episodes at the end of August, as well as an amazing set of events in the fall. If you are an ecosystem member, by now you know that every Thursday you get your weekly news or weekly GPCR news delivered directly in your inbox. If you're not an ecosystem member yet, join us today. Thanks to our fantastic team of volunteers of GPCR scientists, a GPCR article will also hit your inbox weekly on Tuesdays. Also, mark your calendars for not one, but two Dr. GPCR University classes with Dr. Terry Kenakin. The first one is titled Techniques for Effective Lead Optimization of Candidate Molecules, Principles of Pharmacology for Drug Discovery, which will start on Thursdays, September 5th at 10 a.m. EST and run for 4 consecutive weeks for 90 minutes. Then, on October 3rd, an advanced class will start about therapeutic translation of GPCR activity, advanced method for the optimization of candidate selection. This class will run for 5 consecutive weeks, also from 10 a.m. Eastern Time on Thursdays. For both courses, we will be offering 5 scholarships per course to GPCR scientists who live and work in developing countries. So stay tuned. Just a short week after the beginning of our second Dr. GPCR University class in the fall with Dr. Kenakin, on October 11th, we will be hosting our Dr. GPCR symposium on GPCRs as therapeutic targets. During that day, we'll have talks from speakers of all levels, both from industry and academia, a poster session, as well as a panel discussion. We're also excited to be co-organizing our very first in-person Adhesion GPCR workshop in collaboration with Dr. Anthony Boucard from the Adhesion GPCR Consortium. Mark your calendars for October 23 to 25 and join us in Mexico City for a fantastic meeting on all fascinating aspects of adhesion GPCRs and more. If you'd like to sponsor the Adhesion GPCR workshop, please email us at hello@drgpcr.com. Both emails, actually, we have now 2 emails, hello@drgpcr.com and hello@drgpcr.org. Both of these emails will be active for a foreseeable future to make sure that we don't miss your emails. And now, let's dive into this episode. GPCR. Welcome to this new edition of the podcast. Today, I have the pleasure of having with me as a guest Dr. Terry Kenakin. He is currently a professor at the Department of Pharmacology at the University of North Carolina School of Medicine in Chapel Hill. Hi, Terry. It's such a great pleasure to have you with us.
Pleasure to be here.
Thank you so much. It's great. This is a dream come true. As you will hear at a not yet released podcast episode, I was talking to Dr. Brian Airy from BMS, and I was mentioning to him that I really want to get you on the podcast, and here we are. So let's start at the beginning. You're currently a professor of pharmacology. Can you tell us a little bit about your journey to where you are today?
Oh, yeah, sure. I actually started out as a chemist. I'm from Canada, Edmonton, and I got my BS in chemistry. And in fourth year chemistry, realized chemistry is too hard. I then got a PhD in pharmacology in Edmonton, with every intention to return to Canada and teach. I did a postdoc in Britain, London, with Sir James Black, who had just come from industry, and it was who convinced me that industry might be an option. That this was the 70s, and the thought that a scientist would enter industry was rather anathema. You're selling out. So I was all bent on returning to teach. But I remember Jim was a very forthright Scotsman, and he said, "Well, do you want to help people?" And I said, "Oh, yes, of course, Jim. Yeah, of course I do." "Well, then make them drugs when they're sick." And that made sense. So he was the one who convinced me that industry would be a viable career. The other thing that happened after 3 years with him, he became head of Wellcome Trust, and he hired me. so, so I started, and then I transferred to USA Wellcome and Research Triangle Park, and I spent 7 years there. And then the head of Burroughs Wellcome, Pedro Quattrocasas, was invited to open Glaxo in the USA. So he hired me to help him do that, me and others, of course. And so I transferred, and now for the next 25 years, I went through Glaxo Inc., Glaxo Wellcome, GlaxoSmithKline.
Wow.
Iterations, and that's what shaped my thinking in terms of quantitative drug activity and so forth. And then in 2011 I left GlaxoSmithKline and came to UNC, where I am now, and I'm a professor of pharmacology. I still consult with industry quite a bit. I'm privileged to work with Brian Roth here at the UNC, and I sort of head up the graduate student courses here.
Back to teaching.
Yeah.
Back to where you wanted to get back.
Yeah.
You kind of went for full circle there. Here's my question: before you got into chemistry as a young child, I want to say as a teenager, were you how come you were interested in science? Where does that come from?
Very uninteresting story. When you're a kid, you really don't know what's what, and so I basically looked at my school career and realized chemistry was my highest mark, and I seemed to enjoy it, so I entered into chemistry. But I will say, from the minute I got into it in college and science and reading papers and doing all that stuff, it grabbed me. I thought, this is where I'm, meant to be, and I've enjoyed it. I tell students now here at UNC, because think of it, as students, we have one job: to get good marks. So if we get good marks, we're successful. We think we've earned what we should have. And so you almost feel that you're entitled. And I tell the students here, no, research is a privilege. It isn't a right. It isn't that you're smart, because there's a lot of smart people out there. And so it takes more than just being smart. You have to have the desire and the heart for it and so forth. And it is a privilege to do research, I think, anyway.
It is, and you have to be able to move past all the disappointing results that you might get in the lab setting.
No,
and come back the next day.
It was funny, because at GSK we had this quotation on our wall by Winston Churchill, and I'll try to remember it word for word. He said, "Success is the ability to go from failure to failure with no loss of enthusiasm."
Yes, that sums it all up when it comes to working in the lab, or for a lot of things. And so you started out in chemistry, and then you moved on to pharmacology. Where do GPCRs come into play in your
Ah,
career?
well, to step back from that, it's very, very important to say that I was a chemist before I was a biologist, because uh, biologists and chemists think differently. Chemists have rules. They know their universe. They have rules that don't change. NMR couplets, melting points, and so forth. So they know where they are in space. Biologists, hey, our cells were happier today than they were Wednesday. EC50s change and so forth. And chemists hate that. When you're trying to mate those worlds, the chemists hate the vagaries of biology. So it really behooves us biologists to um, make our answers on how active a compound is as chemical as we can. Scales like affinity and efficacy, not potency and so forth. Those things change with the system. So I still think like a chemist. And so I strive to do that. Now, as for the receptors, that was happenstance. I got to the department. They had a young British PhD just joined the department from Oxford, England, who did his PhD with Sir William Peyton, David Cook. So David was, of course, trained in receptors. Peyton was one of the giants in the field. So automatically I was thrust in this world of receptors, which is where the history of pharmacology is, after all, because these were the easy access points. They're on the outside of cells. They give you responses you can see and measure. So a lot of the kinetics and models and so forth that we use were derived from receptor systems. So I just naturally slid into what has been called classical pharmacology of receptors, and found it to be enormously rich and fun.
It is. It is. I think it's beautiful. I always thought that it was very beautiful to go from, as you mentioned, cells that on Monday look one way and on Wednesday they act differently, but then measuring the effects of a drug or multiple compounds on those cells and then translating it into a beautiful curve where you can quantify something. You can put numbers to that state that alters the cells.
Right. No. It's the quantification that really is the important point. And to that, we owe people like Mackay, Rang, Schild, Arians, all the masters. Think of it, with tools as crude as string, sticks, and animal organs, they derive the models, the equations that we still use now. This was really important stuff.
I think so too. And then you had mentioned that. So once you were at Glaxo at GSK, were you still working as a chemist or as a pharmacologist, or were you also using both skills at the same time?
Yeah, no, I can no way claim to be a chemist. Don't ever think that I'm a chemist. I was a chemist when I entered pharmacology, but I was hired as a pharmacologist. And in industry, you come into a department and you sort of I it's odd because you're employed but you're unemployed. You come in and you're not really employed till you have a project. So now you're hunting around, you're trying to be useful, you attend meetings and so forth. And I was basically drafted into a CCR5 program for AIDS, HIV entry. because I was trying to get a program started in orphan screening. So I talked about orphans a lot. And at the time, the receptor was an orphan. Um, so I joined the project and eventually became leader of the project. It was known it wasn't an orphan, it was a chemokine receptor. But the point of all this is it got you involved in a real live screening to chemistry to making a drug project, which is a whole different thing. Then you are employed. And most projects in industry, at least at GSK, give it a year and a half, 2 years. If you don't get any significant progress, it's terminated. But the HIV program continued for like 6 years. And the reason for that was they had a large interest in HIV. They had protease inhibitors, they had compounds already there, and they wanted an entry inhibitor. So this gave us the opportunity to really explore an area, much more so than if they would just go in, screen, and then leave. But the important thing for me was that it gave an allosteric hit. Screen was run, the compound was found, but it was allosteric, and that's a whole new world. That's not your steric hindrance stuff. It's very elegant, very complicated, and I like to say that for the next 2 years we entered the allosteric school of hard knocks. But it was enormously exhilarating but frustrating because of probe dependence and so forth. Compounds do different things, and so the chemists were very
disturbed,
depending on us biologists, and it was a learning curve. But it taught us an enormous amount about allosteric protein function. Allostery is now a huge field. Receptors, so it really opened up things in terms of what we could do.
And this was when, in what year period?
Oh, let me see. It was in the early 90s when it was realized that we had an allosteric hit in HIV. We actually took it to a compound, a Clavaroc, which entered humans. It failed because of, in phase 3, because of liver tox, but it worked. It dropped viral load quite a bit. But since then, I was started and worked on several allosteric projects within industry, which was very useful. Now I consult with companies on allosteric projects because allostery is enormous potential in enormous headaches.
It is. And at that time, when you discovered this compound, that it was an allosteric ligand to CCR5, what was the state of what did people know about allostery in general? Was this a first?
Yeah, good question. Allostery, the enzymologists were years ahead of us. Pharmacology is actually a very conservative pursuit. We're very steric thinking. So at the time, allostery was not considered to be too important because our screens were basically steric screens, binding, and we unveiled steric hindrance type compounds. As soon as the technology improved where we could screen with function, then we started to see more and more allosteric compounds. And as these entered the field exploded. I would add, we only have 2 or 3 approved allosteric drugs, but many, many, many in phase, preclinical, phase 1, 2, 3, coming down. So it is a field exploding in pharmacology.
There are a lot of positive properties of allosteric modulators or allosteric compounds. Where do you see the field of allosteric compounds in the future? Do you think
Well,
what do you
yeah.
think we're missing to get more drugs that are allosteric compounds?
Well, again, step back to we treat allostery now and biased signaling as if it's new, but of course it isn't. It's new to us because we didn't have the tools to fully explore it. But allostery is a basic function of all proteins. So a protein has a shape, and that shape dictates how it interacts with other species, other proteins, and so forth. If that shape changes, the interactions will change. It turns out that the shape dictates the type of interaction. This is called probe dependence. So one of the great things about allostery is a compound can produce a state that causes an interaction with a G protein, and you get something else. A different compound may make a different state and give you a different interaction from the same receptor. This is normal, standard probe dependence, and it is what we call biased signaling now. When it first came out, it was new and rare and so forth. No, anything new is considered rare. It's only rare because you haven't seen it. It's common. I expect a screen to give you a compound that will in some way be biased differently from the natural hormone. I expect it because that's what proteins do.
They do, and sometimes when you read papers or you look at images, you have to remind yourself that actually the proteins are always moving. They're not static, and everything changes around them, whether it's outside the cell or inside the cell, and they react to their environment, whether it's a G protein or a different beta arrestin or different ligands or that come into touch with them.
When we first started to study biased signaling and so forth, there was a tremendous difficulty because the models we had were not capable of understanding it until dynamics entered the field. And you had people like Ongun Onaran, Tommaso Costa. These were pioneers who brought in normal, what the protein chemists have known for years, that proteins move around. There's an ensemble out there, and compounds choose species. But they brought that into pharmacology. You need that to understand probe dependence, because how can you otherwise understand how two compounds can direct signaling to two different pathways other than having dynamics and this open canvas of numbers of states that you can have. So that was a very exciting time. It was started in the late 80s and went through the 90s, and people started talking about it at meetings, and it started as a fringe fanatic fringe, until it was realized everyone started seeing same things. And the key was that we had different assays. We had assays that would monitor G proteins and beta arrestins and different G proteins. As soon as we had the eyes to see, we could see, all compounds are not doing the same thing. Before that, we had no way to know that. We would see the cell respond, but beyond that, not. But once we had the assays, it was quite clear that nature had a much richer array of options than we ever thought.
Agreed. I think the fact that these different assays were developed was really key to discovering all these different states
The
that the
assays,
receptor
yeah.
could adopt. I remember the time, and I haven't been in the field as long, just to say so. And I remember the time where we were measuring G alpha S activity or G alpha I activity by purifying, the cyclic AMP and using columns, and then we switched into plates. And then before that, we were even trying to measure calcium flux in cuvettes with a dye, with a fluorescent dye. But that was really restrictive into looking at different compounds at the same time, and then moving on to with different plates, different assays. I think it's just it just revolutionized the field.
Definitely. Pharmacology in general as a field is really technology driven. You think of all the advances over the years have only happened because we finally had the eyes to see this and then the new eyes to see that and so forth. So it's always changing. It's a field that is enormously dynamic. Papers that are 10 years old are almost useless now because so much more is known. It's very fast moving.
It is fast moving. So you mentioned that at GSK you started out working on the project on CCR5, which you ended up leading. Were there any other GPCRs that you worked on, and do you have a favorite?
Oh yeah. Oh gosh, yes. Oh gosh. Well, chemokine receptors I like. They're pretty complicated, nuanced. We did a MC4R, but I really like GLP-1. The idea here is that GLP-1 mediates insulin secretion. When you eat a meal, you release GLP-1, which potentiates release of insulin. If you have diabetes, this system doesn't work as well. A good way to do it, you activate GLP-1 with something like exenatide. Well, that works, but people feel terrible. They have nausea because you're constantly activating the receptor, and receptors don't like that. So here's a perfect scenario for an allosteric PAM. You have a compound present, it does nothing until you eat a meal. Then it potentiates the natural response, and then it stops when, that response ends. So we had a program in that. It didn't pan out, but I have to say, I've consulted on other programs since then, and there are some active areas. I predict there will be a GLP-1 PAM out there approved someday.
Hopefully. I think it's a very unique system. And you mentioned chemokine receptors. I quote unquote scientifically grew up with chemokine receptors as well, and I think they're a very complex and very fascinating system.
Well, yeah, the thing about them is when we entered the field, just as we were starting to hear about biased signaling and so forth too, chemokine receptors are redundant. We have a tremendous redundancy. There's 20 some odd receptors and 50. They all interact with each other. And at the time, it was thought, this is just fail safe. No, they're all biased. Nature has figured it out. One of the really most impressive papers I ever read was by Trudy Cohout. She was working with Bob Lefkowitz, and she published on CCR7, and she found that CCL19 and CCL21 are the natural chemokines, but they're different. One recruits beta-arrestin and one does not. So nature has actually used this so-called redundant system for fine-tuning. So it really is an interesting textured area now.
I think so too. I think so too. I always found it fascinating that you have this discrepancy in numbers of chemokines versus receptors.
Yeah, yeah.
And then if you look into the human body, the expression changes from cell to cell, and then you don't controlling the expression of the chemokine receptors and the chemokines. That makes it even more complex and beautiful at the same time.
Yes, and having said that, I'm probably not nearly as into this as I should be. The people who do kinetics, people like Sam Hoare and so forth, tell us, oh, you're only seeing snapshots. The kinetics really matters too, and that's probably true. So we're missing a lot unless we start to think about that.
Yes, we are. It's interesting, Lena. I think that was the time to announce this. So Sam will be giving a virtual cafe presentation in August.
Oh, excellent.
Where he will talk to us about quantifying kinetic responses. And we were just discussing, and he had asked me if I wanted to share some of my CCR2 data that we had acquired back in the day when I was doing my PhD. And I was telling him that I did all the beta rest recruitment experiments as a function of time. And because I wanted to know what happens over time with all these chemokines. Mm-hmm. So people will be able to see how we interpreted the data, and if Sam comes to the same conclusion in August. So I invite everyone to take a look at our website and register for their free ticket to Sam's presentation.
I have to tell you a story, a little anecdote.
Of course.
Jim Black's lab in England set up my own lab, and after 2 years returned to England to visit him. And he had just won the Nobel Prize and so forth. So we had dinner, and I was proud. I thought, oh, I've got my own lab set up, and I couldn't wait to tell him about it. And I remember, so I'm telling him what I'm doing. And he says, yes, but do you know anything about kinetics? And I said, no, I don't. Then you know nothing. So that was look at, okay, there's more out there than I thought.
There is, and I think the fact that we were talking about how technology and techniques and the ability to measure the activation of different signaling pathways has improved over the years. I think being able to measure those different signaling paths, the activation of those pathways as a function of time, gives us opens a whole new window into better understanding what these compounds with these natural ligands are doing on in the receptor system.
Right, but I think the challenge, because I review papers as we all do, and there's a lot of papers that come out and say this data will help us understand, and that's great. But for me, the object is to make better drugs, to use that understanding and make a drug out of it. So we have to go the next step beyond understanding and saying, okay, so kinetically this differences occur. How do we use that to make a compound that'll be a better drug? And that seems to be the hard part.
It is the hard part, and I think the audience as well knows, and you and I also know, that being able to characterize differences between compounds in one system, that's great. We got there, but then how do you translate it into humans or into a disease state?
I mean, there's a move now to do phenotypic screening, which is really elegant. I'm all for it. I mean, it's and we have tools now for guys like Fabian Vincent are experts in this, and it's very elegant. But we have to remember pharmacology was built on phenotypic screening. We screened in tissues and animal tissues years ago, and we got phenotypes. And it was only in the later years that we started to associate that with targets. But just being able to quantify this. So my point is, I was asked the question, do you think bias signaling is useful? Because look, one of the first compounds, the angiotensin compound, was found not to be superior in humans, and so forth. And I said, yes, but people have looked at drugs that are known to be good drugs retrospectively and found they're biased. So probably they were good drugs because they were biased. So it is already quantified. So I think we're catching up with the phenotypes, figuring out why they're good, and hopefully that'll aim us towards making even better ones. I don't know. We'll see.
What does it take to have to go from, having a disease state that you want to modulate? Let's think of diabetes, type 2 diabetes, and you want to target a GPCR to help these patients. What does it take? What are the steps? What are the hoops that you have to go through in order to say, well, I think I have a map to find the compound, a good compound that could help? I know it's not an easy question, but I was wondering, since you have the experience not only as a pharmacologist but also as someone who has worked in industry for so many years.
It's an enormously hard question, and you're asking the wrong person. And here's why. I was born and raised in the target-based era. So back then, you would define a target, say, I think we can fix things if we target this enzyme or this receptor. And the whole program was aimed towards that. Whereas disease can be a multifaceted thing, and it may not be just one target and so forth. So now with phenotypic approaches, you actually try to screen and use the most complicated system with all the pieces there already, diseased cells even, if you can. And it's a whole different way to think. You aren't zeroing in on scaffolds and compounds and target activity. You're looking at nuances in the whole complex behavior of systems. I would suggest there are this is a new wave, and I would suggest, if you can interview guys like Fabian, Vincent, and experts in this field, it really is a different way to think. So I would give way to them in terms of your question. How, you have a disease, how would you approach it? Because back when we were doing this, and still now, we would find data that said, well, the CCR5 is the important piece, and we're going to get a compound to modify that. And that's how you'd go. The question is, what if you're wrong?
Yes, I think so too. That's why I was wondering. I was asking the question because when I look at it, it seems to me that looking at the disease in general and trying to understand what happens in that disease state, what goes wrong, and what can we rectify, or what can we modulate, would be one way to look at it. But on the other hand, knowing that we're trying to modulate just one target in a disease state and saying, well, we're going to just hit one receptor, for example, and it's going to solve all our problems, may not be the best way to go for it.
You will read retrospective articles on this. What's best? Target-based, systems-based. Target-based wins all the time because there's many, many, many more of them. I'm not so sure it is the best. An example might be, GSK, but other companies, you would have a tumor, and it overexpresses a certain kinase. So you reason, okay, if I knock out that kinase, I'll knock out tumor. So you make a selective inhibitor of the kinase, it works, knocks out kinase, you put it in the tumor, and the tumor laughs at you. No, I've got other ways around that. And so when you think of systems, you can think of systems as being collections of nodes where pathways meet and so forth. Nodes, and there are redundant ones, which if you knock them out, the system says, I don't care, I can still function. And there are fragile ones. Fragile ones, you knock them out and the whole system crumbles like a house of cards. And so the object should be, we should get to those fragile nodes. What's a key enzyme? What's a key piece that the system cannot do without? And I think phenotypic screening purports to try to do that. They will try to test the whole system and see which ones crumble it, and zero in on what the fragile node is. But again, I'm not an expert in that, and I'm out of my realm there.
The reason I was asking these questions is because it seems to me, and then again, it's a naive perception maybe from the outside, is that it's like playing the lottery whenever you're discovering a compound, because it is hard to define what's a pathway, what is a node that is essential, and what is a node that isn't, or that, as you mentioned, the tumor could laugh back at you and say, well, I can do without it. And in my mind, the drugs that are available on the market, or that were available on the market back in the day, kind of discovered by chance, and it just worked, but we didn't know what the target was and how it worked. What are some ways that you think we could go around this playing the lottery type of drug discovery?
Yeah, a lot of man hours, women hours have been spent in drug companies trying to answer that very question. Because when you commit to a project, you're minimally a year, maybe a year and a half, but an enormous amount of resource. So these are important decisions because you only have a few of them. Within a lifetime of a project or a researcher, you only have a few shots. So, and I've been present in many meetings and discussions and so forth of this very topic, and I don't know of anyone who has a insight that would move field beyond where it is, which is why it's where it is. Except maybe this new move towards phenotypic screening, which I return to again and again only because it, just like biased signaling, it's real now because of the technology. We finally have the technology to explore what it can do. Previously, we didn't. So once you have that technology, it sort of behooves you to exploit it. So I think in the next 5 years, we will see, because with every new field, you will have the enthusiasm, that comes out. This will solve all our problems. And five years later comes the reality. So we will see. So it's too early to see, but I think using new technologies, genomics and so forth, with these complicated systems it's at least going to give us data we've never had before. And so given that, hopefully we can use it to make drugs with it. We'll see.
Hopefully. What do you think? You mentioned phenotypic screening and acquiring new data. where do you think machine learning, AI sits into this realm? How would that be helpful into
Yeah.
identifying those critical nodes?
You know, the short answer is I don't know. I don't know enough about it. You will talk to proponents of AI and they'll say one thing, and you'll talk to critics and they'll say something else. That's normal. But the one drawback that I find with these complicated systems, they require you can't have a human look at this heat map and say, oh, there's the drug. You know, it takes computers and very sophisticated mathematical, analysis. In other words, not everyone can do it. You can't go to your laptop and do this work without technologies like that. So that's the one thing I find limiting. And with that, AI is just another level on that. Who has access to AI? Having said that, we collaborate a lot more than we ever have, the internet and so forth. So I would imagine with experts collaborating, this is doable.
I think I agree with you. I think having the ability to acquire a large amount of data from different sources, whether it's a phenotypic screen, whether it's you're looking at different activation of different signaling pathways, or you will need that computational side of things to get faster to those conclusions, because at some point looking at numbers at a heat map, it's very difficult to come up with the right conclusions. And you were just talking about collaboration, and last year was an exceptionally difficult year for everyone around the world with COVID. How did that change the way you work, and how did that change the way you collaborate?
Yeah. Well, I had a large number of trips canceled, but I mean, with Zoom and Teams and so forth, I'm not so sure. It's just evolved, and I think it's very efficient. It's true, you do miss a bit from the face to face, but I think it's forced us to hone our communication skills as well. When you have to, limit your chatting to a half hour on Zoom, you will be probably more precise than if it's dinner, for 3 hours and so forth. So I'm not so sure it's hindered science that much. But I don't know. A lot comes from having a meeting where all the people meet in a hall, and then, the bar afterwards and so forth. There's a lot of interaction which you don't get now. So I'm not sure. Having said that, I believe we might return back to those days. I don't know when, but
Hopefully we will.
Hopefully.
Hopefully we will. I think based on my experience with Dr. GPCR for the past year or so, I think that people have been very open to holding Zoom meetings, to chatting, to collaborating.
Mm-hmm.
And I think we also miss the ability to go to the bar at the end of a conference day after dinner and just chat up people, or just have a glass of wine and have a discussion at a poster.
Mm-hmm. Mm-hmm.
I've heard a lot of collaborations that came out of just discussions at a meeting just like that. But I think we're resilient enough, and hopefully we'll get back to those meetings and in person, the ability to chat in person with each other.
Right, and it's also eased up schedules. I mean, when you travel, it takes up a chunk. It takes you a week by the time you get on the airplane and all travel. And so now it's an hour Zoom, and you can do 4 of those, 5, 6.
Agreed. I know, and I think people have also become a little bit more accepting of the fact that, in a Zoom meeting you see a cat walk by, or a dog barking, or a dog snoring, or things like that. I think it changed our perspective as well of who we are as professionals and as humans, basically.
Yeah, it's interesting. Yeah, right.
So you mentioned that you consult with industry and you also teach at the same time. How did you get into that consulting phase? How come?
Well, it basically the scientific problems came to me. You get known, people get known in their research area, and people keep up with people. And so it was just, I left GSK, and I could not consult, of course, when I was there. But when I got to UNC, I started to get requests. Would you be interested? And I found that enormously interesting, because as much as I love academia, and I do miss the chemist biologist interactions making drugs, the actual medicinal chemistry, structure, activity, relationships. You don't have that. So being able to go somewhere and take part in a program where that's happening was wonderful. I could enjoy that. So, and it just grew from that. And I found it enormously enjoyable.
It's like basically being in academia, but at the same time still having that first look at what happens in industry when it comes to discovering drugs.
Yes, and I'm enormously fortunate to be in a department with they have they accept this. They say it's all right and do it and so forth. So it's been mutually beneficial. Although without the travel, there's not as much effort. You do more Zooming and so forth, but it's still active.
That's great. That's fantastic. I asked this from each of my guests, and I know the answer to this, and everyone says yes to this without wanting to bias you. But do you think GPCRs are still good drug targets?
Oh, yes. And I scream it, yes. But having lived through the GSK target-based years, when basically GPCRs were thought to be mined out. We've exploited this beyond. With the advent of allosterism and biased signaling, it's complete rebirth. It's just we were just scratching the surface. So now it's an enormously exploding area. The thing that attracted us first to the target probably was they're conveniently located on the outside of cells, so we don't have to get in there for a start, and they control a myriad of signaling. Wow, there's a good start. Now we can fine-tune the way these things signal. So no, there's no question to me that this is it really has given rebirth to the field, when it was thought to be, at one point, it was thought to be mined out.
What do you think is missing when it comes to tools and initiatives that would allow us to improve or increase the number of drugs that are targeting directly GPCRs? What are we missing?
That's a good question, because, I mean,
it's a tough one.
I guess what's really moved the field forward is the eyes to see, having different ways to quantify, things like biosensors. You put these things in a cell and they light up whatever, compounds light up different ones. And you're getting texture now. Instead of the cell responds, no, the cell responds in a certain way. So getting more of that, getting convenient assays. I think 7-transmembrane receptors, which are what GPCRs are, but a better name is probably 7-transmembrane receptor because they interact with more than G proteins. So I think more tools with what these things interact with, beta-arrestin's one, of course, might help us too. They're probably doing more than we think. And so I guess, technologies to further dissect signaling from these guys might be useful. I'm also a big fan, having benefited from it from virtual screening. I think this is a tremendous advance. You get guys like Brian Choiquette in San Francisco just doing miracles, finding drugs out of nothing. So that again was a technology step. You have enormous computing power. So I don't know, let's see. Virtual screening, better dissection and signaling. And then finally, and I don't know how to exploit this one, but we talk about biased signaling. And so you have two assays like GS protein and beta-arrestin. You test 8 compounds and you get 2 groups, one that prefers, one that prefers. You use 6 assays, you'll get 6 groups. And guess what? You use 10, etc., until you get to the point where guys like Stuart Maudsley and Lulu Trell and so forth, you get a genomic fingerprint. Every drug is giving you a different genomic fingerprint. So that's about as detailed as you can get, really teaching us all drugs are different. You can't just say this is a beta receptor agonist. Now, there's nuances beyond that. So I guess being able to exploit that better therapeutically might be useful. Not sure how, but
yeah, that's a tough one. I was just watching Stuart's talk, the Ernest meeting, the European signaling meeting, and he was talking about how environment actually changes your response to drugs.
Absolutely. Oh, good point, Yamina, because there was a Gordon conference about 2 years ago now in California, and we had an evening session on bias. And it was clear that, it's easy to quantify, easy to detect. The hard part is the translation. We take this, we get something that tells us these compounds are different from a simple assay. But when it gets in vivo, the environment completely, further nuances that. And so that's where the drug fails, because if you think, I think I know what it'll do, and now we still don't understand the translation. So the whole evening was spent on how do we do this? And I don't know anything came out of it.
It's a tough question, because it really is. At the end of the day, I feel like it is really dependent on the person. The same, yeah. We were just talking before I hit record about the COVID vaccine, and you had mentioned that two of your children got the vaccine, and one had zero effect on the other one. And when you think about it, they grew up in the same household, but they're 2 different individuals who have different environments.
Yep. And the challenge will be, not every drug works in everybody, but the hope is you get the one that works in most of them, you know?
Exactly.
Yeah.
At least the one that works in most of them to an extent that is enough
Mm-hmm.
for
Yeah.
most people. Agreed.
Yeah.
Agreed on that one.
Well, I've witnessed the change in the atmosphere over 25 years in industry. In the early years, it was like the FDA would say, "Oh, you don't come to us with a drug that's toxic," rightly so. And if it is, we're going to throw it out, to the realization that, you know, you might be able to use that drug in a subset of patients. So don't throw these drugs away because they don't work in everybody. We need to try to help many. There was a drug that um, GSK had, and it was for IBS, and it worked until it really started to give toxic events, and it had to be withdrawn. Fair enough, obvious. But the outcry, the numbers of letters from people who were helped by the drug and then had it withdrawn, you know, was heartbreaking, because doing that hurt as many people as, well, or more than actually having toxic events. So we have to figure out how to use these drugs, they're very complicated tools, better, instead of just saying, well, it should work on everybody, and if not, throw it out. So that's a challenge too.
I love that. I love this idea, and I was just thinking about repurposing drugs, and the example that
Mhm.
came out
Yeah.
of was the AMD 3100 compound, the I can't
Yeah.
the Mozobil. That was the name. That was the commercial name of it.
CR4, yeah.
Yeah, and I thought it was such a nice way of
Mhm.
repurposing, and also making, how to put this in a nice way, actually, and not throwing out all those years of work that scientists put into that,
Yeah.
those chemists that put the work into modifying the molecule, but also those who tested it, and it is a useful drug now.
I heard it explained once that an approved drug is a, you know, wonderful probe. You know, it you can put it into humans and see what else it does, you know.
Exactly.
Quite useful.
Exactly. Under the right circumstances, obviously.
Oh yeah.
with
Yeah.
with the safety, with that minimal safety present.
Mm.
You'd mentioned that, so you've spent a long time in the industry. A lot of people who listen to the podcast are early career young researchers.
Mhm.
If you have any advice for these young career researchers, what would it be?
Well. Yeah, I mean, I now work with graduate students, and they're a lot smarter than we were when we were in school. I'm saying we as the people my age, um, because they're more open. They're industry, academia, they have no prejudices. They just want to practice their science. So I would encourage that. Don't be wary of industry, because, you know, I've heard the criticism. Well, if I go into industry, I won't be free. I won't be able to pursue the ideas I want. They will tell me what to do, and I tell the student, no, they're not going to pay you that amount of money and then tell you what to do. They want you to tell them what to do.
Exactly.
Your job is to bring the world of science in-house. You take part in meetings, you read papers, you become an active scientist in the world, and bring that into industry and try to apply it. So I never felt constrained in industry. I never felt controlled or I couldn't do science because there's so many problems and so forth that, you know, there's lots to do. And you have resources. So your question, I'm sort of meandering off the topic, but to not constrain your thinking that industry and drug discovery is going to be an avenue where you won't be able to do research. You will do very cutting edge research in industry to make drugs. I've also heard it said not to link yourself to a technology. Don't become the imager and that's all you can do. Do a postdoc if you can. I mean, if you have a job offer, take it, But if you have an option to go to a postdoc, do it in a different area, learn something new, and go to the top lab that you can possibly find to do it. So these are kind of well-known ideas. I'm not, adding anything new. That's probably advice I give.
Agreed. I think over the years, the stigma of going to the dark side has
Yes.
dissipated, and I think, as you mentioned, being in industry you're valuable as a scientist. You bring your brain, and yes, you are paid to think. You are paid to work collaboratively. You are paid to innovate, and you have the resources to do it. Because at the end of the day, you're there to come up with a compound. You come up with a drug.
Yes, and of course, subsume your ego. You will be part of a team.
Yes.
Others have said you'll be a cog. But I never had a problem with that because you're working with people who are experts in their field, and that can be enormously fun.
Yeah, and working with equally smart people in different fields, I think that's what makes a team very well rounded up and capable of moving programs forward. And at the end of the day, in industry as well, since it's structured differently than in an academic lab, for example, you will learn new things as well.
Yeah, and timing. When I was looking for a job and just leaving England, this was just after the Vietnam War and so forth, and corporations were viewed with enormous amount of mistrust. Not that they aren't now, but there was a prejudice against it. It was now. As I say, students are smarter. They're much more open-minded, and there isn't that anathema. I would also add, unfortunately, yes, industry has it can be unstable. We've all experienced that. I have. But these days, academia is not so hot either in terms of granting and so forth. It's getting very competitive there too. So the sort of the stigma of industry is insecure is not really that much worse than academia in some ways. But
yeah, I think based on the observation I've been making in the past year or so, I think industry is a fast moving environment. And as you all know, I live in the Boston area, and there are biotech companies growing like mushrooms everywhere, especially in Cambridge. There is no street that doesn't have at least 15, 20 biotechs if you just look around. And sometimes people start, they in one month they work at this company, and a year later they work across the street at a different company. I think the whole point is to be smart, to be an expert at things, to be able to think creatively, and to be able to learn new things. And that that will help you move on from position to position, or within the same company, or from one company to another.
Yep, yep. And size of company really is not that critical. GSK is huge, but they were, you're still in subsets of that huge structure, which are as labile as a small company. So,
Yeah. Yeah.
same
Yeah.
thing. Mhm.
Yeah. Well, I think one main difference between, big companies and smaller companies is, in smaller companies, you may have access more easily to the CSO of the company or to the CEO of the company if you're 30 people compared to 30,000. But at the end of the day, I think it's a great way to take science to the next level using industry resources.
Well, I tell students one of the greatest things about in industry I found was you were closer to the actual endpoint. When we made our HIV drug, I mean, you actually could get feedback from the trials, and it was enormously satisfying to see a compound that you saw from the screen emerge and actually help someone. That was a big kick.
Of course, it's like seeing your baby doing something that you actually wanted it to do. Absolutely. What everyone knows that you wrote a number of papers. Well, I wanted to ask you about writing books. I know you have written books. You have edited new versions of your pharmacology primer book. What does it take to sit down and write a book?
Oh, I wrote the books as an enormously selfish exercise. I wrote it to be able to learn the field. You learn things when you have to teach someone about them, understand it until. So years ago we started with Raven Press. It was a small outfit, and I didn't expect anyone to read it even, but I wrote it just for me. So that was it. And I enjoy writing, and I just kept doing it. When we got to the primer, which is with Elsevier, that's when things started to get more formal, and they've been enormously helpful. They just let me, so I don't stop writing. I've been writing that thing for 20 years. You just keep, because the field is changing. What was known back then is not useful anymore. So it's a field that will always be there to update. I'm currently working on the 6th edition. So, but it's an enormously selfish, thing. I enjoy doing it, and it helps me understand things. If it helps people with their work, great. But,
it does. It does. I can confirm it does.
It isn't Stephen King,
No, but for a pharmacologist, having that as your Bible, as your reference, whenever you're trying to understand, a system, I think it's a really useful book to have around. I hear.
Oh, sorry. There we go.
Not a problem. I have 2 more questions. First one is, do you have any? See, I think the dogs want to talk to us a little bit as well. So, question number 1: do you have 1, 2, or 3 moments, aha moments, that we'd call as a scientist, that make it while you were working in the field, that made you change your view about receptors or about pharmacology in general?
Let me just try to
Not
get.
a problem.
Aha moments. Oh boy. That is a tough question, especially when you're as old as I am. But so there was one when I read the paper by Spengler, who let me just try to. Sorry. Okay. This was in the early days of biased signaling, and they showed that two analogs of PACAP reverse potency with, I think it was G protein signaling and some other pathway, GSGI. There was no way everything I'd been taught in school, right from scratch, could ever explain that. When you come across a finding that just completely cannot gel with what you believe is true. So that was one shaking moment. Another was I was very secure thinking I knew what efficacy was. I had Arians's model and Stephenson's and so forth. And then as soon as you could see drugs had more than one efficacy, it shattered me. I thought, oh no, what are we going to do with this? So, aha, in that you realize your world was too simple. There's much more going on. So, it isn't an aha insight. Oh, now I see. It's a, oh, now I don't see. Those are big ones. And then anything we started to learn in allostery, probe dependence, where you see different findings with CCR5 binding assays versus functional assays. That didn't gel either with what we thought. So those are the aha moments, and I'm afraid they're all aha. I need to study more.
Which is not a bad thing at the end of the day, because it only, what doesn't kill you only makes you better.
Yeah.
That's amazing. And my last question is, before we hit record, you had mentioned the kids, the dogs, the cats. How do you manage, or how did you manage over the years the so-called work and life balance?
Oh, early on, at least in industry, I'm not sure, is you realize it, especially in the 80s. The 80s, if you ever see a movie like Wall Street, if you give 40 hours, that's good. If you give 80 hours, they'll take it, but why not 120 hours? So there's no limit to what you can give to your career. It's up to you. You can give it all, and so I've realized if you do that, then you lose other things. So it's all self-monitored. You say, well, here's where I stop, and I have no idea, where I would have been had I not stopped, where I'd be if I had to stop sooner. You just adjust it until you're happy. So, but definitely these days, back in the 80s, there was a bias towards working an important thing versus now people are a lot more conscious of life quality and kids, family. It's a much more important pursuit than it ever was. So, which is a good thing. Yeah.
it can only make you a better scientist. I
think so. Yeah.
I don't know about you, but I always had the best ideas while I was not working in the lab or not reading a paper, while I was walking the dog or doing something completely
There you
different.
go. Yeah.
Absolutely.
There you go. Yeah.
Thank you so much, Chair, for your time. I really enjoyed our chat.
I enjoyed it. Thank you.
Thank you. Talk to you soon. Bye.
Bye-bye then. All right. Bye. Stay healthy.
I need a favor. Please share the Dr. GPCR University program with your friends and colleagues, especially if they live and work in a developing country. Please subscribe to our YouTube channel. Many of you come back to watch our videos but aren't subscribed. Having more subscribers will help us get you more content. Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest and our Dr. GPCR team, Attila, Ines, Montserrat, Ivana, Cam, John, Yatsu, Andreina, and Balint. If you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. And don't forget to subscribe to our monthly newsletter on our website to get all the news about all the work that we've been doing and we are doing at Dr. GPCR directly delivered into your inbox. Another great way to support us is to share your favorite Dr. GPCR programs with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com or hello@drgpcr.org. As I mentioned, both emails will be active for a foreseeable future to make sure that we don't miss your email. Until next time, stay safe.